Afternoon, everybody. My pleasure to be here, and of course to be in person is great. Wanted to start by taking us back a second to kind of a big picture issue, and something that I've spent maybe the last 20 years thinking about in both starting companies and in my former role as an academic. And it's the issue that much of cell engineering is being driven by exponential technologies. So what are exponential technologies? Exponential technologies are those that increase in time exponentially. So if you think of something like Moore's law in the blue here, this is effectively computational power. So hopefully you all know that that doubles every 18 months. So if you think about the implication of that, the little picture here gives us the implication. It's 2000. We had the Apple computer. In 2010, we had the iPhone. And this little guy, which hopefully many of you are holding, you'll think about this later, is more powerful than the computer, but obviously a lot cheaper, a lot smaller, and a lot more capable. So the same type of technology advances are happening in our space as well. So DNA synthesis is increasing at roughly the same pace as computational power. It's actually a little bit faster, but DNA sequencing has been, 2 to 3 times as fast as computational power. So what does that mean? What that means, to get your head around this, is that every year we're able to, and we do, sequence more than the history of the field combined. That's kind of massive. Every year, more than the history of the world combined in sequencing will happen next year. Okay, so what that does is it means the technologies you think about today, there's a good chance they're not going to be super relevant in 5-ish years, 6 years, 7 years. And that's driven much of the technologies and the methods that we've used to do cell engineering. So that gets us to this part of the slide. If you look at this part, the y-axis here is technical capability. And what that effectively means is how good are we at engineering the cells we engineer. Many of these companies here were started, I think Amyris is 2006, OPX is 2006, so 15 years ago. And these are microbial companies, so they're engineering yeast or E. coli or other microbes, and really throwing synthetic and sequencing capabilities at very similar types of problems that what we're trying to get at in cell therapy engineering. Amyris ended up with a strain with 60 edits in it, and they scaled that. So that went into manufacturing. You're talking hundreds of thousands of liters of producing a strain with dozens upon dozens of edits. A company I formerly operated at and founded, we were building an instrument, which is now on the market, that was selling push button. You can make 10,000 multiplex edits. They're barcoded. You can track them and really harden the ability to do complex editing and engineering in microbial cells. So my co-founder and I and several of the leadership team, we came out of this world. It's kind of hardcore engineering world, where again, we think hard about sequencing and synthesis and how do they advance and what does that mean for the future of the technologies we're going to use, and then how do we put that together into engineering workflows. And we had a lot of friends and colleagues over in this space. And in this space, what we're looking at, the x-axis is market impact. So companies where even a single edit, a single viral edit, randomly inserted edit, can lead to billion-dollar exits and hopefully in the future, many billion-dollar products. So really the founding thesis for Artisan was that we could take this capability, this know-how around how to engineer cells in complex and precise manners, and we could adapt it to some of the cells that had the largest impact. So that's Artisan. And I'm going to tell you a little bit about what does that actually mean in reality. So where are we at today? So we have a proven team of engineers, innovators, and company builders. So from the leadership team, we have founders of companies within the cell engineering space, the CRISPR editing and engineering space, inventors in that space, the cell therapy space, Nick Timmons, our Chief Development Officer, Pete's a public CFO for the last decade or so, and really a fantastic group of advisors helping us think about how do we build out our platform for cell engineering in the best way. So the vision is, as I said on the last slide, we're going to unlock the full potential of cell therapies by building a foundry for cell engineering. So the initial focus of the company, clear focus, is on the engineering platform. So we want to get that right. We're going to start with that, and then we'll think more about indications as we get bigger and better. Founded in 2019, team is about 50 now, spread between the headquarters in Denver and then the development team in Toronto. We focused 2 years on our foundational platforms. So we filed a bunch of IP at this point around editing systems, CRISPR endonucleases, novel cell engineering, and cell therapy targets, and then engineering workflows. We have a bunch of other stuff, but this is kind of the core of what we've protected thus far. I think we've done a fantastic job of validating our platform. So we started off the company with a few collaborations, one with Takeda and Cabaletta and another with Nkardi in the iPSC space. We've delivered on those so far, over-delivered in my opinion. It's really a great team that Tanya and Nick have put together on those collaborations. We have started and are making progress on our own internal portfolio, and I'll talk just briefly about that today. And then as I started with, the team we've assembled, we think is pretty impressive, both within the group of operators that we have, as well as the advisors, both from cell therapies to clinicians to synthetic and systems biology experts. Okay, building a fully integrated engineering platform. So we're building it from discovery or the research phase all the way out to IND enabling. This platform includes working in high-valued cells, so we've got a lot of data so far in T cells, both primary and JERCAT, as well as iPSC and several other cell types that we've worked in. As I mentioned, we have our own engineered CRISPR systems. Any good engineering group knows that you need lots and lots of analytics, so we're building out that platform now, again, going from the analytics, from research all the way up to IND enabling. And then, again, if you've done cell engineering for a long time, you know the data is critical. If you can't understand the data, if you can't collect it, put it together, analyze it, you don't really learn what you need to learn for the next time you do the engineering. In terms of the features we think are enabled by our foundry, we've heard a lot about these. It was a great panel this morning, so we're going after all the same kinds of things that we've heard are the problems. Antigen escape, other types of escape. We're looking at allogeneic modifications, and so on and so forth. Okay, here's probably another linkage slide to put together the larger story. But I'm going to start actually on the right. I'll go from the back to the first. So this is a great slide, took directly from ARM, and we know that if we can engineer cells and we can produce them and manufacture them, we have a massive number of indications we're going to go after. There's a massive impact. So to do this, to get to as many indications as we want, we need medicines that we call intelligent. So what do I mean by intelligent? What I mean by intelligent is effectively the number of decisions or interactions that are going to happen between the medicine and the target. So if you think of a small molecule, that might be only one electrostatic interaction. You get to a biologic or an antibody, maybe that's 5, maybe that's 10. You put that into a cell, and now that interaction is then activating a CAR, which is turning on regulatory things, which then release cytokines and other things. So the more interactions you have, the more intelligent your medicine can be, and ultimately the more of these medical issues you can start to address. To get here, though, to get to these, we need to address these, which means we need to be out here. But to be out here, we need to be over here. And we need to be able to do not just CRISPR. So CRISPR is just a tool. It's a wrench we're going to use. But it's not what you do engineering in and of itself. It's one of the mechanisms we use to do engineering. So what Artisan is about is the engineering piece. Is how do we put together the best CRISPR tools, the best analytical tools, the best workflows to engineer cells in a way that allow us to reliably go after indications that require intelligent medicines. I'm making great speed. How are we building out and distinguishing our engineering platform? So how we're organized is first building the core foundry. So the core foundry, we have it comprised of the research labs, development labs, and cell labs, and effectively going from research up to lead optimization, characterization, kind of early development stages. building our platform here, and again, wanting to go from editing all the way through the analytics. Building out kind of a focus here around novel engineered endonuclease editing systems, methods for novel target discovery, novel targets for cell therapy. So are there different ways in which we may make something allogeneic compared to the traditional ways most people know about? Compositions and methods for enhanced knockout, insertion, and editing. So we have, for example, way enhancers we add that allow our insertion levels to go up. Process and CMC methods. Everything we do is viral-free. So our insertion as well as our knockouts are all virus-free. So we use CRISPR-based RNP methods to edit our cells. And then as we start thinking about our own portfolio, we'll continue to build out the platform and then protect what we come up with. I'll take a second here and just go over where we are in our internal portfolio. So in internal portfolio, we have a couple different partnerships I mentioned. These are all CRISPR engineered. We're not really in a position to disclose much about them, but we're moving into kind of the early, late discovery, early development stages on these. Our internal programs, not surprising, we have blood and solid tumor thoughts, and all of these will be T-cell, allo, and like others we've heard, we're interested in dual CAR and potentially even larger inserts to try to deal with things like antigen escape. We have a lot of expertise in the iPSC space, and we've done a bit of work so far on validating our platform for editing of iPSC. We think there's, as many others do, a lot of potential there. And then we have some hypotheses about additional cell types we might engineer that could be, kind of much longer term solutions. Okay, so I'll go through just a little bit of data here in the last 3 minutes. I didn't have as much time as I thought. This is a count down, not a count up. So now I have to go fast, and if I wasn't already. So data example. So this is what we think about again. I come back to the idea that you've got a lot of engineers who have sat down and built this company, and these go from cell engineers up to process engineering and CMC folks. Engineers love options. So this is an example of our target discovery platform. We can do 1,000 plus precision edits in a week, probably more. I'm being conservative here. This is our favorite target for knocking out TCR in green. This target, we looked at many other sites within that target. We're just showing you one we really like. And this is giving us about 95% functional knockout of TCR. So the issue is, if this doesn't work great for something like you have a serious off-target effect or a translocation event, then what do? And if this is the only target you really like, or it gives you high on target within that particular gene, what are you going to do? So what we have here are 3 additional targets. These are different genes, and then these are different sites within each of those genes that all give us similar levels of high on target editing. So now each one of these we can go look at an off-target assay and think about, use that as a criteria for selection of that on-target site we're going to take forward. So again, it's the idea of bringing people who know about release assays, who know about kind of the end stage of INDE, and saying, what are the targets, what are the assays you think we're going to need in 5 years when sequencing is super cheap, and can we bring those into the discovery stage so we can use those to help us think about what are the guides we should choose now, so that 5 years from now, we're not all of a sudden being asked to sequence something at a depth that we hadn't thought about before. Secondary, in terms of options, this is our STAR nuclease program. All I'm going to say here is we have our own nucleases, and what we've invented is a way to split guide RNA up in various configurations that when they come back together, they give us some control over the ability to do editing, which is showing this here, the ability to dial up and dial down editing at different sites simply by choice of how we split up the guide RNA and the nuclease we choose. Example 2, this is now showing that we are nicely building out multiplex designs. Just kind of hit the top spot here is this is a full allo build where we've knocked out TCR, HLA1, HLA2, and inserted a CAR site specifically and getting CAR expression in the kind of 40 to 50% range. This is showing we can multiplex at as many as 8 different sites and keep, kind of no real loss of the knockout at 98% at 1 up to 98% once we get to 8. Final is just that we're building out the full analytical platform, as I mentioned, going from a few, 40 assays in discovery up to, many, many more as we get further along. And with that, I think I'll stop and show you our pretty video. You can get this from the website. I like the quote because it talks about, again, how do we think of things as engineers? We're trying to make things that work with very few original ideas anymore. So I'd say with that, I'm going to stop, and I have 0 seconds left. Thank you.